make the audience understand why this is important and why some of these need to be considered. Why is there a need essentially to optimize? So this is going to be pretty numbers heavy, but this will definitely set the stage for the next part of the talk, which is the opportunities to optimize. And that's important to understand essentially, what parts of drug development we could use AI potentially, and perhaps what AI cannot do. And of course, there are certain challenges, which brings me to my third part of the talk. Why are we not there yet? And so that will be addressed in the third section. As a disclaimer, this is educational and has helped to build a mental model that you can take back and then you can use on your own. All that I'm presenting today are entirely my thoughts and don't reflect the thoughts of my employee. So essentially, in my talk today, I'm going to show you some of the constraints and bounds in the pharmaceutical and biopharmaceutical industry that will help you understand, what are the levers that you could essentially optimize using AI as tools, right? So no, AI is not replacing our jobs, but it's actually helping us do these jobs better. So I would also like to preface my talk by saying that India has an amazing generics industry. However, my talk today is majorly focused on branded, patented products, which actually have the resources that we put in from an R&D standpoint, or like the cost to patient is relatively higher in comparison to generics. This does not mean that some of these constraints and bounds don't exist in the generics industry. It does. And so hence some of the things that I'm going to talk about could be applicable to the generics industry as well. All right. Okay, so let's start talking about the drug development process or the pipeline itself. So when you look at the drug development process, I mean that we're starting off with an idea. We know what type of therapeutic modality or what type of disease we want to target. We start looking for compounds, right? And this is there's a lot of chemistry, there's a lot of biology that goes into this. At this stage, it's still a compound. It's still a chemical entity. We don't know whether or not it is actually going to be efficacious or not. And so you need to perform a lot of tests, a lot of assays, to transform a compound into a medicine, right? When you're doing this, you're still in the preclinical stages. That means that you have not started testing in humans yet. And so to do that, we perform these clinical trials, which is considered. Sorry, is there a question? Okay, perhaps not. So, in order to do that, there are these different phases of clinical trials where we are actually testing out, these candidate medicines in humans. And this is important because ultimately, these medicines are for humans. And so we need to understand whether, A, the drug is not toxic, and then B, that it is indeed efficacious. And then C, we want to understand what are the side effects, right? Oftentimes, some of the medicines that we take, there can be some side effects as well. So we need to understand that in the context of human biology. And so that proof of concept happens during these clinical trials. And then ultimately, or rather in each of these stages, you have to show the regulatory agencies, so, be it the FDA or the EMA in the EU, about, whether you can produce the medicine at, X quantities under high quality. And so that is that regulatory proof that, we need to keep up with as we are going from one phase to another. Once, we have provided the regulatory proof to these agencies, we are okay to essentially market these drugs in different regions of the world. And that's where you have this last phase of development where you have a lot of post marketing commitments or life cycle management activities. Maybe you want to improve the type of patient administration. Maybe you want to introduce a device. So that kind of research essentially happens later in stage. Now, if you look at this entire pipeline, it takes around 12 to 15 years. And this is average. I'm going to give you some numbers which, may be really hard to swallow later on in the presentation today. But on an average, this entire process takes over a decade, right? So it's really slow, which means that there is a lot of opportunity for us to essentially utilize AI to expedite, to make those mistakes faster and to learn from them faster so that we can restrict this time and we can really squeeze this time, improve the resources that we are putting into this drug development process. So a lot of opportunities here. When you look at the drug discovery and development funnel, in terms of the number of compounds, here also there is a lot of opportunity, right? Because when you start off with basic research, you have a lot of compounds, right? You have at that idea stage for a specific target, there can be several molecules that you can potentially use for that, right? In this case, like, a 5-digit number, so 20,000 molecules. This is where you perform a lot of in silico screening, right? So it's not experimental. It is done computationally. You're utilizing, several in silico tools to understand whether or not this is the correct compound or not. Then you perform target molecule validation, right? You are trying to see whether the compounds that you've generated using, your computational tools, whether they actually are relevant to, the molecule or the target that you're trying to look for or not. So this is where a lot of like experiments come into picture, because you want to validate whether what you're getting from, computational analysis actually makes sense. And then you have a lot of in vivo, in vitro testing. So when I say that, I mean like, you have cells, you test out whether your compounds work in cells, and then you test out whether your compounds work in animals. And this needs to correlate, right? Again, we have not entered human testing yet, right? All this is happening in preclinical setting. So we are building a lot from these models and trying to extrapolate whether this information that we are gathering that could be used for humans or not. You're also performing safety testing, right? You Before you go into humans, you want to know and have some confidence that what type of safety implications could happen if you start administering this in humans. This entire process of just finding out what molecule, maybe performing some experiments for validation and in vitro in vivo testing and safety testing can take about 7 years. So that's a really long period of time, and you've not even started your clinical trials yet. Once you do that, there are different phases of clinical trials, as I mentioned, and each serve a different purpose. For example, phase one clinical trials for most drugs, you're testing out for, what's the safe dose. Generally, this is administered in healthy volunteers, so you've not started testing in patients yet. And then you're also looking at the metabolism. How is the drug getting metabolized within the body? Then you have the phase two clinical trials where you're trying to understand what's your therapeutic window. So what's the minimum and maximum dose that you can administer? And then finally, you have phase three clinical trial where you are, expanding the number of patients. So increasing the N number in terms of statistics to understand, what are the side effects, or how efficacious can the drug be in different parts of the world, from a genotypic standpoint or an epigenetic standpoint. And this process is about a decade long. And then give or take a year to registration process to actually go to the authorities and be like, hey, we have this data, and we feel confident that we can produce this drug at a high scale for our patients. So can you please give us, regulatory approval? And again, the reason I keep saying regulatory approval is this is a highly regulated field, because ultimately, humans are going to use this. And this is unlike, something like tech, where, this would look like in a year you can ship out, a software update. This is not as easy as that, right? So it's a highly regulated field, but that doesn't mean that there is no opportunity for tools like AI to essentially help condense, this, for example, the number of compounds quickly so that we can get to the final product sooner. Now, let me paint another dire picture. When it comes to probability of success, with each phase of clinical trial, your probability of success is actually really low. So many drugs, they actually go through phase one clinical trials, and between phase one and phase two, you have that dip, so many drugs actually die when they are in phase 2 clinical trials. Because in phase 1 clinical trials, you're looking for toxicity, and in phase 2 is when you start looking at efficacy, right? So think about it. Why is that the case, right? And with phase 3 clinical trials, because you have established some efficacy from phase 2, now you're expanding. Now you're looking at, like, statistically, you're seeing whether or not your drug actually can be used around the world. So more often than not, your probability of success for phase 3 clinical trials is generally higher, right? However, overall, for a drug to go from phase 1 to approval, the probability of success is right around 10%. And if you notice this graph here, it's actually been pretty consistent for a decade, right? And this is really low, because like only this basically means the probability of only 10% of your drugs actually making it, to the approval stage. So that's actually very meager, and there's a lot of opportunities here again, where you can use tools like AI to essentially give you a sense of, how can you make a phase 1 drug go through, this cliff of phase 2, where most of the drugs, they die, right? It's called the graveyard of medicines, right? And then it's also very different from an indication standpoint. For example, oncology has the lowest rate of probability of success, right? Not all oncology drugs, like, they basically make it to the very end. And that is sad, because, you start with oncology research, you start in vivo models, right? You start in rats and mice. And there's this joke in the industry that, we've probably tried to solve cancer in rats and mice, but not yet in humans. And you can see that the data here is not promising either. So there is, again, a lot of work, specifically in indications, which we still don't understand, because the biology is so complex, right? So a lot of scope for us to essentially use these tools to, optimize it. Also in terms of modalities, right? The molecule types as well. The years in clinical development is also contingent on what types of molecules you're using and what types of therapeutic classes, because that would again dictate how much of clinical trials do you need. For example, when you're looking at rare diseases, you're probably not looking for a very big patient population. However, so to get a drug for rare disease, your N number, to test out in humans is going to be fairly smaller. However, for something like oncology, it can take a really long period of time, because, what happens as a repercussion of some of these, like, say, for example, chemotherapeutics, right? They could have side effects. And so you wouldn't know of these side effects until very late. So your time that you take to, test out these drugs in patients, that increases substantially. And so again, this is just to reiterate that the time that it takes for clinical development is dependent on not only the therapeutic class, but also the molecule type. Each of these molecules are very different from each other. And so it is not, you cannot use a one size fits all. You could start with a small molecule, which has a specific compound, and, like, synthesizing that in the lab and then trying to scale up. But then as you start with, like, say, a protein, now you're talking about, how do you extract proteins out of live cells? So that totally changes the game. And perhaps the models that you had used to do your analysis, or even scale up and process development for small molecules, may no longer apply to proteins, right? So it's not a one size fits all. Next, I want to introduce you to something called Eroom's law, which is Moore's law backwards. And I want to touch a little bit about on this, because, we talk about, AI in tech quite often. And in tech, we have this Moore's law, which suggests, the productivity improves, or almost, I believe, doubles I think every 10 years or so. But in R&D, in pharma, it's the opposite. We see that there is a decline in productivity with as the years pass. So the amount of money that we are spending in R&D costs actually keeps increasing as you keep going forward, right? And this could be because of several things, right? We are going into essentially trying to develop complex biologics, or, really complex modalities. And so the biology is not catching up, right? It's more evasive. It's less, you cannot code biology still, right? So there is a lot of scope again here where, there's a lot of work that's currently being done. We are not there yet. And I think this is a restraint or a constraint of our field. The reason that we are so slow, just as I mentioned earlier in the previous slide, there is a low probability of success overall across clinical trials, but there's also reduced investment in early development. So if you look at the industry right now, with every passing year, you're seeing that the investment that the companies are putting into early development is actually declining, right? And what is this to say is that, we are not necessarily, not many companies are looking into discovery just because think about it, you're going from like 20,000 molecules to 500, and that's actually very costly. So how where AI can be helpful is, can it help us essentially reduce that number to begin with quite considerably. Now here I would like to point out that artificial intelligence, like I said, is not a magic pill, right? It's not going to tell us like overnight, this is the molecule that is actually going to help you cure cancer. What, because it's not a deterministic system, right? It will not tell us like, answer A or B is correct. It won't do that. It's a probabilistic system. It'll tell us with a high probability of success, I think A is the right answer, right? So it's always going to be probability based, which means that you always need to validate it experimentally to understand whether or not the suggestions that artificial intelligence is making actually is right or not. And this is extremely important because what it can help us with is decision making. So early on in our development, it can actually tell us whether or not a drug has a high probability of success if it sort of benchmarks it on trained data based on what has been already approved in the industry, and has been approved by regulatory authorities. So again, it can help with decision making. So, which is extremely important, as you can tell, it's such a long, like your feedback loop of a cycle is like, around a decade long. You don't want to wait for a decade to know whether or not your drug is going to work. And so this is where, AI can really help us. So why do we need to optimize this, right? I gave you really, very dire statistics before this. And a little bit more on this is the expense in the US, for example, to, get a drug out, at least in 2023, was around $2.3 billion. That's a lot of money, right? And it actually keeps increasing, right? Again, the ERUM's law, right? So it keeps on increasing with every year. The return on investment is not good either, as you can see here, right? Like in 22, it was just 1.2%, which is not great for investments, right? And in 23, it was 4.1%. Again, not the greatest. But the most important reason is that the biggest losers in this whole, game, the whole pipeline is actually the patients. In the US, for example, for newly approved drugs, the cost to patient was around $150,000 a year. To give you an example, in India, when Zolgensma, which is again, it's a gene therapy drug for patients less than two years, and this is a survival drug. Like the infants need this in order to survive if they have, I think it's a spinal injury or a spinal defect, a genetic defect. So kids need this, otherwise they will not survive. The cost of this drug in India is between 16 to 18 crores. So you think about it, if it is so expensive to develop a drug and the return of investment is not high, who is paying the cost really, right? It's the patient, it's coming to us. And this is why there is a need to optimize ultimately so that patients can get access to drugs faster and at a much reasonable cost. Of course, there's a lot of like, policies and things like that. But from my standpoint, I think again, coming from R&D and not from a policy, side of things, there is a lot that we can do to essentially make better use of resources to develop good quality drugs faster for patients. So with this, we go to the next section of the talk, which is what are the opportunities to optimize. So when I mentioned about the different phases of development, I think of it to categorize into three buckets, which I think could be ways in which we can really use AI to optimize and reduce the costs, right? The first one is discovery and preclinical studies, right? This is the place where you have highest attrition. If you recall the slide from before, you have a lot of compounds at that stage, and then you're sort of going down this funnel to get your final compound. So what if you make better predictions early on, such that you're reducing the number of experiments, you're failing fast, or you're failing fast, and that you're getting to a drug sooner, right? So this is where, and also, like, sort of testing it in vitro, so on cells, or in vivo, which is in animals, in a way that you can confidently reduce the tests that you do in humans, right? Run the clinical trials. So this is where there's a lot of opportunity for the use of AI. Manufacturing and scale up. So because I work in this part, in the manufacturing and scale up aspects, I think that there is this is a very engineering problem that we can solve using AI, right? Because again, optimization, right? So as you go from one scale to another, you understand the process really well, and so you can use these constraints to boil it down to a very simple optimization problem that you could use AI. Again, AI will not work if you don't have a lot of data, right? AI cannot work if you just have three experiments. You can run Excel, with that, right? You don't need AI for that, right? AI will help when you start generating a lot of data. So when you're going from, small scale to medium scale, you start analyzing the samples more, and you try taking samples from after every unit operation, and then you start putting that into AI, and then AI can make these analysis that you and I on Excel cannot, because it can look at higher dimensional correlations between factors, and so it improves your process understanding multifold, right? And then finally, there is clinical studies, which again, I think there is a lot of work again that could be done in the way we design clinical studies, such that we can improve the outcomes, right? Safely, of course. And since my level of expertise lies in, mainly in the manufacturing and scale up, but I do have some idea of how it works preclinically, I'm going to focus on these two for today's talk. However, I do think that there is a lot of there is a lot that we can do to expedite that clinical studies as we progress from phase 1 to phase 3, because again, this is a population problem, it's a statistics problem, and you're generating a lot of data. When you're running clinical trials, you're taking a lot of patient data, which you know not necessarily at least previously was not necessarily valued as much. However, now with AI, the processing computing power has improved substantially, which means that you can make use of this data to actually create those go no go decisions sooner. All right. So again, when I think about preclinical studies, I think about this valley of death, right? And while there are many ways, even in the preclinical phase stage, that you could essentially optimize, I think if you look at the statistics, again, you will realize, and the data, you will realize that between academia, which is where basic science research takes place, and the industry where you are starting to test in humans, many compounds fail. And this is because there is this you don't have a testing mechanism, essentially. So like I said, the example that I gave for cancer, we are able to solve cancer in rodents, but not yet in humans. And this is because many drugs, they die when they are getting translated from in vivo or animals into humans, right? So one way that we can overcome this is by utilizing models. And this will help us to understand our molecules soon, because we want to know whether the molecule is toxic, whether the molecule is efficacious, whether molecules can be synthesized reproducibly, and whether or not these models that we are using, that they are biomimetic, which means that they are as close to humans as possible. And so there is a lot of, like, hardware aspect of it as well that is now upcoming in preclinical screening, like the work by the group, where they have essentially looked at several of these models which mimic certain organs, for example. So you have organ on chip, or you have these tissue diffusion chambers, which will give you an understanding of diffusion across the intestines as you consume a drug. And this is important because the research that they're doing this is essentially mimicking how does a lung, for example, in this case, can be modeled on a chip. And so you put your drug on this chip, and you see how it is getting metabolized and affected onto lung cells in this area, right? So I think as the technology is advancing from a device standpoint, or like a model standpoint, an assay standpoint, this is where AI will come in to sort of help use this data, because you're generating data again, right? So you can use this data using AI to essentially understand, whether or not this drug is going to be successful in getting translated from, an academic setting to something which the industry, would deem appropriate and worth. And there are several other ways in which, like in tools, again, use AI as tools that can help to accelerate drug discovery. So here is a non-exhaustive list, essentially, that can really help us to start our research. And I would encourage, everyone here to, use AI and not be scared of it, because this is a good starting point. I wouldn't say that, I would completely rely on it for my research. But this is a good starting point, especially if I'm not familiar with a specific topic, right? I would still go ahead and read the papers. For example, like, with literature and evidence discovery, AI could what we call hallucinate. So it could, give, something which doesn't make sense. But this is a good starting point to give you like a list of resources that you can then go and do your own research. Um, all of this, again, it'll give you starting steps, but you have to experimentally validate it. And this is why I don't think AI is going to take our jobs, just because there will be a human component to understand whether or not the the prompts that we are, first of all, giving, and the answers that we're getting from AI, whether they are actually making sense or not. So I wouldn't blindly trust whatever, like, these GPT models, for example, they're giving us the responses. But I would actually go and like read papers, or like experimentally validate some of the factors that AI is telling us. You may have learned, you may be aware of AlphaFold and the Nobel Prize that the team got to understand the structure of proteins and protein folding, which is a really big problem and a big deal in the field, because we did not understand, like, how proteins get folded. And now you have a model. So imagine, like, the model was built on basic chemistry and physics and biology, and can now tell you how proteins are folded. And as humans, we didn't know this. So the implication of something like a model like this is, now you can find diseases that people are that are debilitating in nature, and you can find therapeutic interventions or therapeutic drugs for these misfolded proteins, right? So again, like I said, I think these are great tools which really help and supplement research, but in no way, shape, or form this is going to replace the work that scientists are doing in the lab. Um, the next topic, which is again more of what I do on a day-to-day basis, it falls under this thing called chemistry, manufacturing, and controls, or CMC. So in CMC, we are looking at the actual compound, which is a therapeutic, which we think is has potential of treating or curing a disease. That is called a drug substance. We transform this drug substance, which is just a molecule, it could be a powder, into a drug product, which could be an injectable or a tablet. That's called a drug product, what patients get administered. And while we are doing this transformation, or even like synthesizing, the drug substance itself, we utilize analytics to understand whether what we are making is what we intended to do in the first place. So think of analytics as like our eyes and ears, essentially, as we are manufacturing drug substance and drug product, and we are transforming one to the other. And so I put them in one category. And while we are doing this, we also have this regulatory aspect, so, and we have like quality assurance and quality control, which again makes sure that we are making the substance, or making, again, drug substance or drug product to the highest quality, and that we are very certain about for example, the stability of a drug product, that which will determine the shelf life of, like, how, for the expiry dates that we have on medicines, they actually have a reason, and they come from some of these quality control analysis and stability analysis. So there is a lot of work that goes into determining the date, the best use by so and so, or it expires on so and so. And this is where, like, you have quality assurance and quality controls, which again police these these aspects of the drug development process. And then you have regulatory. So this basically means everything, all the research that you have done, all the development that you have done in synthesizing a drug substance, scaling it, synthesizing a drug product and scaling it, and the analytics around it, and the quality assurance. You need to give that package to regulatory authorities, and then they are the ones who will tell that, yes, this is a good quality product, and we approve, and you can now market it to patients. And of course, there is like a clinical component to it as well. So, but when I talk about CMC, again, I'm talking about, like, purely the manufacturing aspects, or the synthesis aspects, essentially, of the whole thing. All right. So with CMC, I think there is a lot of scope for us to essentially use AI tools. For example, again, as an optimization problem, how do you use, how can you optimize the yield of your drug substance? For example, can you improve reactor speeds for monoclonal antibodies, which is again a biologics type of drug substance? Can you improve the way you are synthesizing tablets, or you're manufacturing tablets? Can you improve the way that your tablets are dissolving appropriately in your intestines, right? So all this, again, you could utilize several engineering approaches. It's an optimization problem at the very, crux of it. So that could be, that is an opportunity for us to use AI. We could use platforms. So while the different modalities are very different from each other, as I mentioned earlier, your small molecules could be very different from, like, if you have biologics or monoclonal antibodies. However, within small molecules, if you have a tablet, and if that, say, a molecule has a specific property, the drug substance has specific properties, you can actually establish a platform that can help you, drastically reduce the time that it takes you to manufacture tablets. So say you have a platform, and it's called a platform technology. So say you have a platform technology, and this is again built on models, built on a lot of data that you've collected from, like, several iterations. And you understand, using AI, the correlation between, the properties of drug substance and the process parameters. You could actually, as when you get a new molecule the next time, you can use these platforms to essentially go through and get your tablet in record time, right? And then there is this in-process analytics that will help you ensure control. So again, very chemical engineering, in the sense that you need to impose control in your process, and you use analytical techniques like chromatography, or even something like pH assessment, right, or dissolution characteristics, to ensure that with the drug product, the drug substance that you're manufacturing, actually has really high quality, and it is what it's supposed to be. On a regulatory, quality assurance and quality control standpoint, I think that some of these could be automated quite a bit, because these are less dynamic. They don't change a lot, right? Every manufacturing process has to go through quality control. Now, what type of quality control, that of course depends on, the type of molecule that you are synthesizing. However, that remains common. The standards remain common because you have several different pharmacopoeias. However, the interpretation of pharmacopoeias may be different. And so, as long as your model codes for it, you could potentially automate the whole process of, like, getting this information, putting them into regulatory filings, and essentially, pushing them from one phase of development to another. However, as I say all of this, and I recognize that there is a lot of opportunity for us to utilize AI and machine learning. Um, as I mentioned earlier, this is a highly regulated process, and the reason is because patient safety is of utmost importance. So, if we make any errors, any errors in this whole process with automation or like quality control, that could directly impact how patients essentially react to these medicines, and we don't want that. So it is while this is less dynamic, and you can, it's more controllable utilizing AI. You can get a lot of data that you can help to understand the different correlations between the different factors in your manufacturing process. It is highly risky because patient safety is ultimately important. Um, and in the same vein as earlier, here are some of the examples of AI and ML tools that could be used within CMC. So, for example, you could utilize formulation and process development. I know that there is a talk about digital twins later. So, digital twins is also something that we are considering and we are utilizing within manufacturing. And this can really help us, again, generate a lot of data, which can improve our process understanding, right? So we can have simulations essentially of our process. And we could also like monitor using some of these digital twins. PAT and multivariate modeling, again, really important from a prediction standpoint, because like this will again help us to understand, what's happening in the process, and then we can use this information to create a feedback loop. PAT technologies are extremely important for continuous manufacturing. So while continuous manufacturing is very common in other parts of the chemical engineering industry, in pharma it's only up and coming now. And while it has made great progress with small molecules and tablets, for biologics, which is again like monoclonal antibodies, we rely on cells. We are still in its infancy, and it's up and coming. But something like a PAT tool really helps us. Again, this is process analytics testing. This helps us to understand whether your process is being run properly or not. And then you can essentially have a feedback loop to tell how to change the process parameters. So imagine the amount of control that needs to go in and the amount of process understanding that we need to do so that we can make this work. Um, there are a lot of other workflows or another AI and ML tools that could be used. So I encourage you to take a look at these. Again, specific for CMC, specific for the pharma and biopharma industry. All right, so now we come to our final topic of the day. So, this is some of the challenges with AI and the drug development process. So, why are we not there yet? Why hasn't AI essentially developed a magic pill for us, and why are we not there yet? Um, so, the first thing is that there is still a lot of skepticism around AI. So AI needs to start building trust. And we don't know yet whether we are in this AI bubble, whether it is just a fad. We know that, like, for example, if you are a startup company and you say that you're working with AI, you get a lot of funding. People don't ask you how are you utilizing AI? If you just have AI in your pitch decks, you do get a lot of money. And so there is a lot of understanding that still needs to come to understand whether or not AI is actually doing something or not. So is it fab or is it just a fad? And there is also a bit of a confusion of, like, when you say that, okay, so I'm going to use AI, for example, to help me pick diseases, like I'm going to use an AI doctor. I don't know how many of us are going to be comfortable with that, right? We still want real world doctors. And so for the industry to actually make great progress, I feel like AI needs to build trust. Again, this is the pharmaceutical industry, because it's highly regulated. You have patient safety on the line. There is a very big aspect of building trust in the industry. So we need to perform more experiments. Maybe start with low hanging fruits. For example, something like a generics market is a great way in which we can incorporate AI. Just very quickly, when you have generics, it's based off of branded products, right? So you already know what your endpoints look like, and all you need to do is essentially optimize them, make it slightly different so that you can sell it as a generics product. um, that's where AI could be useful, right? Because like here you do not have that much of profit margins, but you still want to improve your process. And then you already have a lot of data, which you can utilize to build these models essentially. So that could be a first step that will also help us basically build trust as an industry in AI. The next one is the understanding of AI. So many people in their regulatory filings, for example, have claimed that they've used AI in some of the different aspects of their drug development process, and there is a bit of a confusion as to what exactly did they use AI for, right? Because like I said, some of the analysis you could also do on Excel. So what are they using AI for? Like, if you could do the analysis on Excel, if they've not generated enough data, how could you train a model, to use AI, right? So people have misused it quite a bit, and that needs to potentially change. There are a lot of companies now collaborating with tech companies to incorporate AI in the workflows. But we still don't have a truly AI generated product, and I will be actually pretty excited to see that change sooner than later. But again, this is like overused, and so people need to essentially take a step back and really ask questions. Okay, what exactly is AI doing? So there is healthy skepticism, but then that also stems by perhaps not understanding what AI can and cannot do. And so I think one of the biggest challenges that we are facing is the lack of data, or rather the lack of good quality data. There is this statement in the field of AI ML, which is very famous, and that is garbage in, garbage out. So if you give garbage data to the models, you train the models on garbage data, you're going to get garbage results out. And that's not going to be useful for anything that you do. And so, apart from that, there are also several aspects in the in a very competitive pharmaceutical industry. For example, all the data that companies generate, these are proprietary. And so, they try to, because this is how they distinguish themselves from, their competitors. And so, there are now consortiums coming in where, especially for clinical studies, right? Because like that's ultimately what, your number of people on the earth are limited. And so, you need, the biology is so complicated that, you could probably really use some of these consortiums where you have companies, depositing their data, maybe even blinding it so that, you can improve your understanding of patient data better. So, but this is only just happening, and people are still, a little sensitive about utilizing data for this purpose. The next thing is quality. You mentioned garbage in, garbage out. You need to have good quality data. When I was preparing this presentation, I was trying to look for some statistics on some of the things available in different parts of the world, and I utilized that there is a disparity in the amount of data that we collect, or the quality of data that we collect, to make any meaningful associations or correlations out of it, right? So when your teachers, for example, told you to make sure that your experiments are well documented, that directly correlates to good quality data. So it starts from there. Like when we are running experiments in the Can everyone see me, hear me?
Yeah, yeah. In
Yes,
between,
yes.
Okay.
your
Okay.
call was interrupted. Yeah. Now you are
Okay.
audible. Yes, yes.
Okay, awesome. Thank you. Um, The next reason for, like, why we expect good quality data is because you want your models to be reproducible. So all the experiments that you're running, you want to sort of repeat them, right? If I'm running an experiment, I would like, the same, and I create, like results which could potentially get me a Nobel Prize, I would want like other people to be able to reproduce that as well, right? So reproducibility is a great aspect, a very important aspect in our field, and so again, directly proportional to the quality of data, all the information that you're putting in, and this is also applicable when we are generating models for AI and machine learning. Data diversity is another big problem, especially like in our industry, because we are looking at different types of data, right? Like, so they could be these factors that can affect the quality of data. For example, if I'm working in a lab, which is temperature controlled in this part of the world, and if somebody else is working, the same experiment in another lab, we need to know whether or not temperatures or humidity have an impact on the experiments or not. And we wouldn't be able to do that, unless we run the same experiments, which would be again considered reproducible. The other aspect in clinical trials also is, when you're performing clinical trials, say in the US, and you're performing clinical trials in India, you could have patient populations which would have different, genetic makeup, but also could have different epigenetics based on the environment. And so your clinical trials need to consider, this diversity so that you can again put these factors in. AI cannot tell you, like, a separate factor which it hasn't accounted for. So you need to tell AI, you need to train your model on data and on factors that you think might have an impact. And so you need to have a diverse set of data so that you can train your models on that to make it more effective. And then finally, and I again would want to stress on this, experimental validation is extremely important. AI will just tell you this is, there is a high probability that you will find success in this molecule if you, do run this synthesis. But you wouldn't know that unless you actually run that synthesis or not, right? Again, this is based on probabilistic modeling on the back end. And so, there has to be experimental verification, which again, you put this data, you make the machine learn, right, from this data that you're putting in. And so experimental validation is extremely important. Every time that you are using AI for anything, you should validate it either by reading the paper completely from start to finish or by performing an experiment in the lab. So in my mind, I think AI could be utilized in every step of the development process, right? Right from discovery, into CMC and even for clinical trials. And there is a lot of scope for us to essentially condense, this timeline, this really preposterous timeline of like, a decade and a half really to a very small period of time. The real question is, how are we going to do this? But I can assure you that we are getting there slowly but surely. So with that, I would like to thank everyone for taking the time to attend my talk and would like to open the floor for questions.
Yeah, thank you, Kanika, for this insightful session. And I think there are a few questions which are posted in the chat box. But before I read those questions, I will take this opportunity to start this question answer session with my own question. So while using this artificial intelligence in drug development, is there any risk to miss out any important step which we generally follow in our traditional drug development process, which may create any harm to the patient in the future? And if so, then what care we need to take while applying this AI in the drug development process?
I think that's a great question because, oftentimes, I think there are, like, in my mind, at least with the people that I've spoken with, I feel like there are two groups of people, right? One, they feel scared of AI because they don't understand it, and they feel like, they don't want to use AI at all because, they fear it, right? And then there is this other group of people who, they use AI for everything, so much to the extent that, like, their critical thinking is at risk now, right? Because they're using AI to make every decision for them. And I think that, we probably need to be in the middle somewhere, right? So we need to use AI as a tool, but we need to, have healthy skepticism around it. So trust but verify. I wouldn't start with AI, for the entire drug development process, because that could miss some, instinctive things. For example, if I'm running an experiment in the lab, and because I have done that experiment several times, I may have some tips and tricks to, like, say, get a reading right, like, do some analysis correctly, just because of my experience of, like, hands-on experience of working on that experiment in the lab. AI won't have that. And so, which is why I feel like, these things could be missed. So, from some details, essentially, if you've done this step, several times, you may not have that quality in AI. And so I would recommend, again, like, utilizing AI as a tool and not as a replacement to critical thinking is going to be extremely important. Otherwise, we are going to miss out on, many of these nuances, which, would actually be critical to develop drugs.
Right. Thanks, Kanika. So let me read out few questions here which are posted in the chat box. So the audience, one of the audience is asking how we can use AI in unit operations. I don't think whether it is directly concerned with your topic of presentation, but as most of the audience are with the background of chemical engineering, and
Yeah.
we all apply many unit operation during the drug development also. So
Yeah.
maybe the audience is asking this question.
Sure, yeah, absolutely. So I can give you an example with one of the processes that I'm developing right now, or the drug for drug products. It's a nanoparticle process, or nanoparticle manufacturing, and one of the unit operations that we are using is evaporation, because we have to get rid of this organic solvent in the system, right? So we're all familiar with evaporation as a unit operation. We have an evaporator, and you have different temperatures, and we have this condenser based system, because we can't heat it really high, because otherwise our protein essentially will degrade. So now you can start thinking of it as, like, you can actually have these equations based on, like, what pressure to use, what temperatures to use, what's my feed flow rate going to be like, what my output flow rate is going to be like, and then essentially controlling that process, by using temperature sensors, using pressure gauges, for example, or if you're using a stirrer, what's my stirring speed going to be like. So in this case, you are going to start generating a lot of data using these sensors which are in process. And when you put this information, again, and what I'm telling you right now, this is very limited information, so I'm not sure if this is the best example for AI. But when you start looking at this, like, for multiple experiments where you have different types of feeds, and you're looking at, like, different outputs, and you put this information into AI, you'll start analyzing, or you'll start understanding what correlations, so for example, in my case, it's a polymeric nanoparticle. So whether or not, the molecular weight of my polymer has an impact on how quickly, will it solidify after evaporation, right? So those kind of answers, you would have to probably run like 1,000 experiments, before you get to the stage of, like, actually having a correlation between molecular weight and, maybe the rate of evaporation, or the rate of, in this case, hardening, right? However, you could utilize AI to reduce those experiments from 1,000 to, like, maybe 20. So this is why AI will help you reduce the number of experiments. You cannot completely negate it, but you will reduce it quite considerably. So it all starts with, again, those equations that we are learning, in different unit operations, mass transfer, heat transfer, and you translate that into these models, essentially, and then you utilize the data that you're generating from these models to perform correlation analysis using AI. I hope that answers your question.
Okay. There is one another question from Mr. Ronak. Uh, does AI helps in modeling?
Um, yes, because so it's a feedback loop. That's what I would think of, right? So in so AI, essentially, you need to feed data to AI, right? And you need to essentially let the model know. So you have an algorithm, which is built on artificial intelligence. Again, this like neural networks and so on and so forth. So that helps you like on the back end. You know, your models, you can essentially use your models to generate data, which again, like, trains the model itself to improve its accuracy, right? So again, AI is used for analysis. And so, like, based on like the correlations and, that you are getting from performing experiments on your models, you can actually use that for improving your accuracy of your model. So yes, that's one way in which, at least in my experience, that we can utilize AI to improve modeling. On the other hand, you could also, like, the other way to think of this is like, say you want to start modeling something from scratch, right? I think GPT, for example, is a great way to do that, right? I'm not sure if most of you are familiar with something called vibe coding, where you're actually utilizing, these GPT models, so AI GPT models like Chat GPT, or Claude AI, to help you code, right? So you can essentially now use these GPT models that can help you code or create models from scratch. And then you can use, of course, you need to validate it, but then you can basically, again, use this to generate data, which helps, make it stronger and stronger, essentially, or more accurate in its in determining your outputs. So yes, definitely, there are many ways in which AI could be used to not only generate models, but also to improve its accuracy.
Fine. Another question is from Maithili Khamkar, that how can we design hybrid AI first principles models that simultaneously optimize emissions across all three scopes?
Um, is this specific? So can you explain what does emissions mean and the scopes? Do you mean
I
like
think Maithili can uh, ask more
Prasad sir,
about it. Yes,
Prasad sir,
Maithili.
that question was for previous session. That was
Acha,
for
okay.
previous question session. Yes.
Okay, okay, okay.
Hello.
Sorry. Yeah. So,
Sir, I have
okay.
one
So
question.
all these questions are from for previous sessions, I think, Trupti madam.
Sir,
Yes,
Bhagavati.
yes,
No, sir. No,
Am I
sir.
yes, sir.
audible?
Yes,
Sir, am I
Ramesh sir.
audible? Ah,
Yes, yes, you are
Madam,
audible.
I have one question. Is there any AI tool is available considering any raw medicine with the some specific diseases that gives the mechanism how is targeting that particular illness or something? Is any AI tool is available nowadays?
do you mean like, for example, if you have like, say, a paracetamol, can it tell you what's the mechanism of action? Is that what your question is?
Yeah. Supposed to be, I
Okay.
consider any raw data is there. Raw medicine is there. Supposed to be any raw conventional medicine that goes to the body and having the very complex mechanism of the body. Then
Mhm.
supposed to be some illness is there. Then can any tool is predict how they react or how the result can predict it?
So this is actually something. It's a part of IVIC or PKPB modeling, and again, it started off with modeling, but now, with more data that you're generating, you can certainly use AI, right? Like, AI is just like data analysis on steroids. So that can actually tell you. It can tell you how. So this is called, like, pharmacokinetics, pharmacodynamics. It is how your drug is interacting with different parts of your body, and then how is the drug getting distributed in the body. What's the metabolism? How is the drug getting metabolized by your body? So yes, there are tools. For example, GastroPlus is a great tool that can tell you, at like, when you take a pill in, or a tablet, right? It will, it will have some, like, say, the absorption happens in the small intestines. And so on its surface, it's going to have, like, an acid resistant coating, which will not disintegrate. The tablet will not disintegrate in the stomach, but it'll wait till the pH actually rises in the small intestine, and then it'll start disintegrating. So that disintegration and dissolution and then absorption into the bloodstream from the small intestine, that can be modeled, and that has been modeled. And so it can predict, what's the mechanism of action, where does this go. Now, to maybe the other way of answering your question is also if you have a molecule, how do you know if it is getting, what's the mechanism of action like? So generally, it's the other way around. You have a mechanism of action, you know that you want to inhibit a specific protein, for example, and that triggers what type of molecules or what type of compounds do you want to discover. So typically, it's the other way around, but I would imagine that, you know, if you have a molecule and if you understand where that molecule, what's the mechanism of action, then you could actually back calculate that as well. So yeah, but as I said, generally it's like you have the mechanism of action and then you're trying to find what compounds would be most suitable for that.
Okay, fine. So there is one another question from Maithili Khamkar. How can pharma companies design validation frameworks that allow early AI-driven decisions without risking patient safety or regulatory compliance? Also, how can AI make sense of the vast variety of molecules in the initial drug development stages?
I think that's a great question because, with the regulatory framework, so again, for me, so we have, every pharma company has a department where they're doing, regulatory submissions, and they are very familiar with the different pharmacopoeias, and they're very familiar with different regulatory authorities as well. But their decision making is driven by the work that R&D is doing, right? So the experiments that we are running in the lab. Now there needs to be this correlation between the work that the experiments that we are running, and whether or not what's the probability of regulatory success, right? So that part needs to be answered by the regulatory team. However, generating data, like I said, in one of my slides, I talk about this, this translational graveyard, right? Where many drugs, they die. So if you can essentially come up with, models which are biomimetic, then you can essentially use that for making decisions sooner. Whether AI can help or cannot, again, I don't think it will give us a deterministic answer of a yes or a no, but it will tell us, that the probability that this drug will work, given the data that you have, provided to the model. Given this data, I think that the drug is going to be, say 40% successful. Some of the ways in which the drug can improve its success rate is by doing so and so, right? Now, whether or not, you implement that and, it gives you high level of success, only data will tell, right? So, but it'll help inform what types of experiments that you can learn, essentially. And then in terms of, I think your next question was on, performing compound search in the discovery stage. So yes, AI has been used a lot in drug discovery. As we are improving our understanding of biological models, and we are improving our understanding of target and ligand associations, right? It's almost like a lock and key. So AI is helping us find the right key to the lock, right? And again, because this is, based on, different compounds and what actually makes sense, it's much easier for small molecules because they are compound based. So something like, paracetamol based compound or something. What I'm trying to say is that, what you synthesize versus something like a biologic, which cells secrete. So that's a little more difficult to do. However, AI is helping us, in finding these, essentially keys to these therapeutic locks.
Okay, there is one another question. What are the chances of errors in drug test and how AI will assist in preventing false positive testing in drugs?
That's another good question, right? So there are errors all the time, right? And so I think, from a purely, mathematical and engineering viewpoint, right? One way that you can prevent errors is by, if you think that, maybe there is an assay that you're running, right? That it it's not a very good assay. Like it does not tell you, for example, if you're running an assay based on like, maybe a chromatographic assay to tell you the concentration of, say a protein, just giving you an example. And that assay, you think that the variability, the standard deviation of your assay is pretty wide, right? So what you would, and such that you could make errors, right? So what would you do? You'd basically look, okay, so I want to determine concentration using chromatography. Are there any other ways that I can use to essentially get me the concentration of proteins? Well, yes, there is. Like, run UV, right? So Beer-Lambert's law, if you're familiar with, right? So you use UV absorbance as a way to essentially get you the same data. So when you start doing this, you start building like, orthogonal testing into your process. And that gives you more confidence that the data that you're getting is higher. I would imagine where AI could be used would be, or like if there are any errors in like, the processing itself, right? Like, maybe something was fed incorrectly, right? But you need to have analytics to tell you that, that this was fed incorrectly. And then like you could have a feedback loop essentially to tell you, to course correct, right? AI, like I said, could only be helpful if you have generated a lot of data to tell you, give you some sort of correlation. But you will need to you will need to train that, the model, the algorithm on what is right and what is wrong, which means that you will have to perform some experiments. So again, don't think of AI as like, a one-stop solution for everything. It is not, it will not give you a solution. It is not deterministic. It will only give you like a probabilistic solution. And it is very much dependent on, on the data that you put in. I hope that answers your question.
Okay, there is one more question, Kanika. So you explained this question. Actually, it's not there in the chat box, but the person has personally messaged me that you explained the general procedure of the drug development process. But during the COVID-19 pandemic, how did this process take place? During that situation, I believe there was very limited data available. If so, is AI really useful in such situations?
So that's a great question, right? Because during COVID, yes, there was limited data available. However, the two companies here that were working on the COVID vaccine, so BioNTech, Pfizer, and Moderna, they were actually working. They were working on the platform. So as I mentioned, in one of my slides about like, the platform technology, right? Lipid nanoparticles as a platform, that technology was already established. So all they had to do essentially is to generate, once they got the protein sequence of, the COVID-19, the spike protein, they essentially transformed that into RNA. Right. So now think about it, right? You have a protein. So again, I don't want to go into the biologics of it. Happy to connect offline on this. But once you have a protein, you can actually, go backwards and see what type of RNA can make that protein. And these vaccines that, Pfizer and Moderna developed, these were mRNA vaccines, right? So mRNA codes for a protein, right? So once they got the sequence of the protein, they back calculated to see what is the mRNA that could give this protein. And they used the platform technology that was already developed to put these two together. Like the drug substance was the mRNA, LNP was the drug product, and then they basically, put it out. So they were actually able to use, some of these like computational tools to transform the protein to mRNA in a record time. So these are some of the situations or some of the places that like AI could be very useful in providing the library of like what are the potential candidates. So now instead of like screening like say 20,000 molecules or 20,000 mRNA, you're probably just screening 5 of them, right? And I did read a book on this, right? And that's what they say, that they had very few formulations that they were then testing on healthy volunteers to see whether or not they're able to improve the titer values in vaccines. So you could certainly use AI in that aspect, right? When it comes to manufacturing, I think that was the bottleneck during COVID, just because there were very few industries that were well equipped with LNP manufacturing. right? mRNA manufacturing. So that was a bottleneck, and that's why it took a while, to sort of scale up. There were the shelf life was limited and so on and so forth. But that was also transformed. Pfizer did a great job in essentially converting some of its converting some of its manufacturing sites into COVID vaccine manufacturing sites. Again, because they did not have a lot of data, I don't think that they used AI for this. This was like, completely brute force, in performing experiments and learning iteratively. But I would have imagined that, like, by the end of it, they must have generated enough data to then give them confidence that, okay, you know what, you can now successfully scale up, this process. So in maybe the next vaccine that they develop, they can use this, in the manufacturing process, they can use this as a template to sort of expedite the scale up process quite considerably. So I would imagine that the the place that they could have or they would have used AI perhaps was in transforming that protein into the mRNA, because that part, you can do some computational work there. But I don't think that it was used in manufacturing. However, as you start generating more data, that's when AI could be of use.
Okay. Kanika, actually, your topic is too much interesting for our participants. So we are receiving so many questions. I hope you are not running out of the time. Then I can ask you a few more questions.
Yeah, go for it. Yes.
Yeah. Okay. So actually, there is one follow-up question for the COVID vaccine only. How do we ensure the safety of human health and avoid risks? Because there have been several cases where the vaccines have a reverse effect on individual like cardiac arrest and diabetes.
So I wouldn't say reverse effects. So those are side effects, and this is going to be a challenge with any new modality that we explore, right? Um, that was actually one of the, I guess, like one of the negatives as well of the mRNA vaccine, because this was the first mRNA vaccine of its kind. That was again, I'm guessing that you're talking about the mRNA vaccine and perhaps maybe not the viral vaccine, or maybe the viral vaccine. I'm not sure, but typically there is this side effects analysis that is done, right? For any type of drug, you need to understand, like, what are the side effects. Now when you talk about, like, LNP and mRNA vaccines, there have been cases of, like, some sort of cardiac events that have occurred, even with I believe even with Spivaks, which is, I believe, a viral vaccine. You again have a similar thing that you see, right? So unfortunately, there is no way to know this unless, like, you start gathering data to see, what exactly caused it. Was it related to some of the comorbidities that the patient had, right? Was the patient already, like, was the patient predisposed into having, cardiovascular disorders, right? Did the patient have high cholesterol, for example, that worsened the patient's, reaction to these vaccines? So this is a part, like I said, this is a very important part of clinical trials to understand, side effects of medicines in general. And so that should not be taken lightly. Oftentimes you wouldn't even know if, there is a side effect. So for example, with cancer, right? With oncology, you could have these drugs that can actually potentially could change your DNA, and those changes you wouldn't know in a very short period of time. You have to wait for a long time. And so as that data keeps coming, that's a part of, like, the post marketing commitment that industry makes, that when whatever data we get from the patients that are using our drugs, we're going to take that seriously. We're going to take that as a feedback loop and maybe put that as a warning label on our the packaging insert that goes into our drugs. So side effects is an important part of clinical trials, and also post marketing pharmacovigilance as well. But it could be because of many reasons. That may not be in the scope of especially post marketing commitments.
Okay, Kanika. So actually, I'm very happy to mention that all these questions and queries, they are coming from our own present students. So they are very
Oh, that's nice.
enthusiastic to know. Yeah. So they are very enthusiastic to know many things related to this session. But considering the time limitation and we have one more presentation after yours, so I will request the participants and those who have many queries and doubts to connect directly with Dr. Dr. Kanika later on, so that you can have a conversation with her with your queries and questions later on. So let me conclude this session by delivering the vote of thanks. So it was very nice to learn how this process of drug discovery pipeline can be accelerated by using this artificial intelligence, how we can accelerate the literature survey, evidence discovery, how we can speed up the process of identifying the molecular structure, new molecules, etc. So it was very insightful session that we learned here in this, during your presentation. So I wanted to extend my heartfelt thanks to Dr. Kanika for sharing her expertise on the application of AI in the drug development pipeline. Your insights on how AI is revolutionizing this field were very truly enlightening for all of us. The various examples you shared on AI-driven molecule discovery and this clinical trial optimization, they were very much fascinating for all of us. So thanks again, Dr. Kanika, for inspiring all of us with your talk. Thank you very much, Kanika.
Thanks everyone. Thanks. It was lovely, coming back to TSEC and talking about this. Thank you so much. Thanks everyone.
Sure.
Take care. Bye.
Yeah, thank you.
Thank you very much.
Okay, so let's start with the next session. Ms. Sharvari, are you here in the meeting?
Yes, sir.
Okay. So let me take 2 minutes to introduce our next speaker. Yeah. So I welcome Ms. Sharvari Kulkarni to deliver her presentation for the upcoming session. So let me take this opportunity to introduce Ms. Sharvari Kulkarni. She is a Chief Technology Officer at BDG Life Science. She is working there as a research scientist and CAD expert. She's pursuing her PhD. Her expertise in the area of bioinformatics, especially in basics and advanced bioinformatics, drug design, molecular simulations, and dynamics, and mathematical modeling and network pharmacology. She has more than 15 international publications in this domain, and she has also helped more than 50 PhD students in their projects in the field of CAD. She is recently honored with an award of one of the 10 best women leaders in the pharmaceutical and life science industry 2025. With her article published in International Women Entrepreneurship magazine, she has trained thousands of participants with a track record of more than 90% positive feedback. Her major strength is able to create well-structured lesson plans and engaging learning materials that facilitate active learning. She is one of the favorite trainers of the participants, reason being her patience and teaching methods, and troubleshooting of errors with personal attention to each participant of the workshop. She is able to communicate effectively and clearly, adapting her communication style to suit differently learning styles and abilities. Her sessions are always interactive with the focus on understanding and developing interest for the topic. She expertise in the field of computer-aided drug designing with her proficiency of operating on molecular docking and MD simulations on Windows as well as Linux operating systems, with her training programs ranking second with 7 stars. She can perform docking of all types of biomolecules with vast data sets and their visualization in different visualization software. She can use multiple software for validation and comparative study with in-depth data interpretation and guidance in publication. She has excellent command on performing MD simulations on Schrodinger, Desmond Maestro, as well as Chromax platform, which is challenging and key skill in computational biology. She is the only trainer in CAD who conducts training on molecular dynamics simulation of protein ligand complex simulation with hands-on case study exercise, which includes setting up the environment and standardizing the protocol for complex systems. SHARVARI has prepared universal codes for performing in-depth analysis of MD simulation on GROMAX. She guides research projects for college students on various innovative topics in computational biology. She is a renowned international speaker on various topics of computational biology. So it was a brief introduction to Ms. SHARVARI Kulkarni. And once again, I request Ms. SHARVARI Kulkarni to deliver a presentation. Over to you, SHARVARI.
Thank you so much, sir. Good evening, everyone. And thank you so much, Prasad, sir, for my kind introduction. And I sincerely thank the organizers for inviting me to become a part of this faculty development program and for giving me this wonderful opportunity to interact with you all. I am actually ex-teacher of Thadoma Shahani College of Engineering. So there is always a special connect because it was my first workplace. And now, as mentioned, I'm working in corporate. I shifted from academics to BDG Life Sciences, where my job is to conduct trainings. So now I'll start sharing my screen and we will quickly start with the topic. Okay. So I'll start first with a brief about what is drug designing. Sir, just let me know if my presentation is clearly visible and audio is also.
Yes,
Okay.
yes, ma'am.
Thank you so much
It's
for
visible.
confirming. Thank you so much for confirming. Yeah. As Kanika has already explained us about the drug designing, I wouldn't go into the basics too much, but since we are going to talk about AI and ML in drug discovery, I need to just build your foundation because I cannot jump directly to the topic. So we will gradually stepwise see where actually we can understand the contribution of AI in drug designing. For that, it is essential for me to talk about the highlighting points about the drug designing. So to define it in simple words, drug designing is the scientific process of discovering and developing new medicines that can treat diseases by interacting with specific biological targets, such as proteins, enzymes, DNA, or receptors in the body. Now, if you ask me, how do you compare or how do you understand? Many a times people now that AI has entered into drug discovery, but we don't know what actually is the difference between when you say conventional drug designing, when you say CADD, and when you say AI and CADD. So I'll give you a very simple analogy. First, let us understand the difference. Conventional drug design relies on trial and error. CADD uses computers to guide the decisions, and AI in CADD enables system to learn, predict, and design smarter drugs, right? So I give very simple analogy of how they can be differentiated. So you are, suppose, take an example of analogy of walking to an unknown destination without a map. It's the conventional drug design. Riding a car or bicycle using the Google map is what is CADD. And driving self-driven car with the already known knowledge of the route, and it will select the best route, avoid traffic, and it is improvised with every trip. That is what is AI in CADD. Now, is it clear? So this is how I would like to define AI in drug discovery. So if you are, it is just like a self-driven car. So you are speeding up the process. You are moving more towards accuracy. But how? By giving the patterns. So this is exactly how we are implying AI in drug designing. Now, let us go ahead with the conventional drug designing. I wouldn't spend much time on this, but this is how you define conventional drug designing. First, you need to identify the disease. Then you isolate the protein involved in the disease, which takes around 2 to 5 years. Then finding a drug effective against a disease, around 2 to 5 years. Preclinical testing, scaling up, formulation, filing IND, NDA, FDA. This whole process takes up around 10 to 25 years. Isn't it a long time? Do we afford that much time in research? Certainly no. So we moved our strategy to CADD. As the name itself says, computer-aided drug designing means aid of computer. So as you all know, this is bioinformatics or CADD is not a very new field. It is old, but I can see I'm into this field when I started teaching bioinformatics to TSEC itself. It was just limited 10 to 15 years ago. So it was just limited to have a theoretical knowledge, not much practical implementation, at least in India. But I can see its drastic and remarkable changes or the speed of awareness of the CADD post-COVID. So we all know how bad it was in the COVID. Wet labs were getting shut. And we, in fact, needed any methodology to speed up the process of research. And that was a game changer to computational biology in terms of CADD. So first bioinformatics vaccine, which was developed with the help of computer, was purely in silico vaccine done on one single computer by Moderna company. It was first mRNA vaccine. And that was the time when researchers started understanding, yes, it is possible to use only the computer and the knowledge of computer to perform research efficiently in drug designing. And that is where CADD was introduced. So CADD represents the computational methods and resources that are used to facilitate the design and discovery of new therapeutic solutions. So of course, the advantages of the conventional becomes disadvantages of the conventional becomes advantages of CADD. So this is a journey where you started with conventional, then CADD, and now AI in CADD. So let us understand how the progress was done. So if we talk about the advantages of CADD, of course, the time is reduced, lesser than half. Cost is reduced, accuracy is more, information about the disease is already coming from the data mining or databases. So the screening is reduced and less manpower is required. So there are two main types of drug designing that we talk about, LBDD and SPDD. So there are entirely two different approaches when you say drug designing. So when you say types of drug designing LBDD, it is entirely based on the knowledge of the structure of the molecule. So we call it often as a lead, as a ligand, unless it qualifies all the criteria of the drug designing pipeline, it wouldn't be called as a drug, right? So till then, we will be calling it as a ligand. So based on the properties of the ligands, when you build a model and you do drug designing, it is called as LBDD, which is QSR, that we are going to see today more in detail, how ML and AI has contributed in LBDD. So it is entirely based upon the properties. We call it in our language as descriptors. Another approach, which is very conventionally very old and it has been used efficiently, SBDD. So when you know the receptor structure properly and the designing is based on the structure of the receptor or the target, in our case we call it as a target on which the ligands are supposed to act and inhibit the disease, it is called as SBDD. Now we will see section wise how AI is contributing to LBDD and how AI can be contributed to SBDD. So when I'm saying I'm starting with SBDD, because we are going to see only the tools which are used in SBDD in terms of docking and in terms of MD, but we are going to see demo of how the tools can be used for LBDD. So it should go in the flow. So I'm taking SBDD first. So main steps in drug designing are target identification. So when you identify a biological target involved in the disease. Second, lead identification. Find molecules that show activity against the target. Lead optimization, which is for modification of the lead to improve its selectivity, its activity. Now, let us start from very basic. When you say a drug, what criteria or what qualities you expect from the drug? It has to be active and very important, it has to be selective in its action, right? So lead optimization is improvising on these two things. Admit properties. Admit properties is absorption, distribution, metabolism, and excretion properties. So this is another important thing. Unless you qualify, be it a very good ligand in terms of docking, in terms of MD, in terms of LBDD, if it does not qualify admit analysis, you cannot take it further for the clinical trials. And of course, the toxicity is concerned. So you have to also see the tools which can predict the toxicity. And then it will go for preclinical trials, then the routine thing, right? Preclinical trial, clinical trial, lab test, animal studies, and human trials. When we say SBDD, it uses entirely the knowledge of the 3D structure of the target protein. So the common techniques are molecular docking, molecular dynamic simulation. Example, designing inhibitors using the crystal structure of enzyme. When I say 3D structure in terms of PDB for docking or MD, it comes from the X-ray crystallography data, right, in terms of 3D coordinates. And that can be taken further as a receptor or target in your MD and docking studies. So I'll give you one real world example. When I started also on this kind of SBDD projects in CADD, I'm saying, what kind of data do we deal with? So when I'm handling this kind of projects from big companies, research labs. So I'll start with an example which you can relate. So when we started in the COVID, you have COVID receptor 5, right? The membrane protein, spike protein in terms of PDB. And we used to always hear that you should drink turmeric milk in COVID. Why? Because it was hypothesized that turmeric has some extracts which are chemicals which are inhibitors of the COVID. So what we do in such case? Client or the scientist will come with the data where you have your library of ligands. So you can take that from the database also. We have different databases like Impact, we have PubChem, we have Drug Bank. We take the data of the ligands and then we perform docking on that. So I deal with as small as the data of 10 compounds to 1 lakh molecules. So we perform on Linux the docking studies. We shortlist from huge data to smaller data by performing docking study. Conventional or authentic tools are Autodog, Vina, Autodog 4, Pyrex. Then when you scrutinize or shortlist, which we call as a virtual screening, from this you scratch down only to 10 compounds. This is called as identification of compounds. And then you take it further because till 4 or 5 years before, we used to get satisfied with molecular docking and it was understood to be complete study. But now we don't trust on docking. We have to go one step ahead, that is MD simulation. So why are we doing all these tasks? In order to see the stability of this protein drug complex inside the natural environment, that means in the body. And for that, you have to perform MD simulation. What is the meaning of simulation? Simulation means to mimic the behavior. So we are giving the environment which is exactly similar to our natural environment, and we will see with the help of different parameters, like when we analyze the simulation, we see its trajectory in terms of RMSD, RMSF, SASA, radius of gyration, energy contributions, MMPBSA analysis. All this report, combined report of docking and MD will qualify it as a drug. So if I started with 500 ligands against COVID, I will short list only 10. And after MD, I will qualify only 2 to 3. And from that, again, it will go to Swiss ADME. So this is the beauty and efficiency of CADD. Instead of synthesizing it in the wet lab, you already have its report or prediction which of this is going to act well. Only that will be synthesized into your wet lab. This is called as CADD. Okay. When we say LBDD approach, LBDD is when you do not have any knowledge of the target. So it relies entirely on the known active compounds. So the techniques for LBDD are QSAR, which is quantitative structure activity relationship, and pharmacophore modeling. Most commonly and efficiently used is QSAR. What is the role of computer in it? Of course, to predict the affinity, to screen the 1,000 compounds virtually, and to save the time, cost, and lab effort. Very successful examples are like to target the cancer-related enzymes, design small molecular inhibitor. Of course, it will block the enzyme and stop the cancer cell growth. Now I would like to give a concept of docking so that you understand what are we actually talking about. Then I will give you one slide explanation of what is MD, and then we will talk about its tools. Okay. So when I say docking, docking is an attempt to find the best matching between two molecules. So I always give this analogy. It is like a lock and key. So finding the right key for the lock is nothing but your docking. What you're doing in docking, you are placing a ligand into the binding site of a receptor in the manner appropriate for optimal interaction with a receptor. So we are optimizing the interaction between ligand and drug. And then with the help of docking protocols, you are evaluating these interactions in such a way that it discriminates the experimentally observed mode from others and estimate the binding affinity. So this is a very simple slide I always put in order to make you understand clearly what is docking. Here one man is opening the lock with the help of key, right? So if the lock and key has complementarity in the shape, it will get open. not only the complementarity, but also the direction in which your key is moving. That is also important, isn't it? If you move it in the reverse direction, will it open? No. That is exactly the case of protein and ligand. So there are some spots on the protein, which we often call as hot spots or active sites or the grid box. These spots are the target spots on which ligand will go and bind, and also it will bind in a definite or a proper mode. If it binds to that mode, in that mode, then only you will get the best binding affinity. So your score will be maximum and you will get the best possible interaction. So what is our objective in the docking is to optimize this interaction between protein and ligand in terms of complementarity in the charge and shape. So for example, here you see this is the protein, this is a ligand. For a better view, I have just zoomed it like this. This is the protein and green color is ligand. Ligand is making interactions with protein and you can see the labels and the number of amino acids with which it is making interactions. These amino acids we report and that are essential from your interaction point of view for the protein drug complex formation, right? So when you perform autodog vena docking in the file of the, when you execute the command, you will get a file with 9 modes of results as the top ranking first one with the least docking affinity at the end. You visualize them with the help of Autodoc tools, PyMol or Discovery Studio. And if you have seen papers related to docking or MD, you must have seen these DSV images where the interaction study has been done. Now, out of this, suppose you have sets of ligands, the ligand with the best mode and best possible interactions will be taken further for MD. What is MD? MD is a computer simulation method for analyzing physical movements of atoms and molecules. So whenever you want to study any system, how do you judge the system? Of course, on the basis of any biological system, how can you judge the stability of the system? By studying the physical movements of atoms and molecules. So here, when MD is performed, it is the calculation of the time-dependent behavior of molecular system. So it provides detailed information on the fluctuations and conformational changes of proteins and nucleic acids. So it is routinely used to study the complexes, protein drug complexes in terms of this stability. So what is the standard protocol in CADD? First, you perform your protein docking study, you get a complex, and then that protein drug complex is taken forward in the MD studies. Mathematically, if you want to define MD in simple words, it is nothing but generation of atomic trajectories of systems of n particles by numerical integration of Newton's equation of motion for specific interatomic potential with IC and BC. Okay. ICBC means initial condition and boundary condition. Now, what are the platforms and tools for MD? Docking, I perform means people perform docking in computational biology on Windows or Linux. There are different platforms which can be used. Autodoc Vena and Pyrex are the most important tools that you can use. If you have less less molecules to be screened, you can go for Windows. And if you have large molecules to be screened, then like I told you, I do work, keep on working on as huge data sets as 1 lakh, 2 lakh molecules, right? So in that, I use batch code for on the Linux platform. Okay. Then Pyrex is also one automated software which is used for docking. MD, you also perform it nowadays on VirtualBox, which is subsystem for Linux, and you can perform on pure Linux if you have partitioning. Gromax is freeware on which you can perform MD. And AI-based is Desmond Schrodinger Maestro, in which you do not have to perform any coding. You simply load your protein and ligand and you run it for 100 nanosecond, 200 nanosecond, which is the criteria of publication of the paper. Depending on that, you get a report, PDF report, just like your blood report, all the parameters. So everything comes with everything comes for a price, right? So if you have money, I advise you to go for Desmond because its license is very costly. It is for an year, it is around 2 lakhs to 2.5 lakhs. But Gromax is entirely free. Only the thing is you should have good patience with the codings and you should have that much, you should be prepared for doing hard work and patience. Then you are good for Gromax. Okay, so it depends on what is your system also, what is your purpose also, and what is your like choice, if you want to put efforts or you want to put money. Then how AI and ML can be applied to this. So broad concept of AI is nothing but mimicking human intelligence. You agree with me, right? And machine learning is how are you going to mimic the intelligence? By giving the algorithms that predict the data or that learn the patterns to predict the data. Why AI in drug discovery? So of course, the same thing which I have said, the period or the time is cut down, cost is cut down, and improvisation on the activity and toxicity in the early stage. So in these steps, AI fits into drug discovery. You can use it in target identification. You can use it in hit discovery. You can use it in lead optimization. You can use it in admit. You can use it in drug repurposing. Now I'm going to start with very important topic of the today's session. I'm going to highlight on AI and ML in LVDT, and we will learn how to make mathematical model for the drugs by using only their properties. Okay, so QSAR and ML we are going to study. Let us first understand what is QSAR. QSAR is quantitative structure activity relationship. So it is essentially the relationship between chemical descriptors and biological activity. How do we say biological activity means? IC50. Why I have written here PIC50 is because in QSAR we prefer to work on PIC50 because it builds good model. PIC50 is negative logarithm of IC50. So it fits well for the normalization of the data. Equation in general terms is activity is F of descriptors. We are going to do 1D, 2D, and same is applied to 3D also nowadays. So it is depending upon which descriptors you are using. It is called as 2D descriptor or 3D descriptor. So accordingly, it will be called as 2D QSAR and 3D QSAR. So basically, QSAR is a computational or statistical method which establishes a quantitative relationship between the chemical structure of molecules and their biological activity or physicochemical properties. Core idea in QSAR is conversion of molecular features in terms of structure or properties into numerical values. Okay, these numerical values we call as molecular descriptors. One can build mathematical model predicting how active a compound will be based on these numerical values. So biological activity is a function of molecular descriptors plus, of course, since it is a mathematical model, you have to also consider the error margin. Now, conceptual roots of this QSAR, they are traced back to 19th century with the only notion that molecular structure have the influence on the biological or chemical activity. It is often summarized as the broader concept of SAR, structure activity relationship. We move ahead in one step as a QSAR with the formalization. So instead of just qualitative SAR, QSAR tries to quantify the relationship, enabling the prediction rather than mere observation. In SAR, you are just observing. In QSAR, you are quantifying it so that you are coming up with some important predictions. So over the time, QSAR has evolved. As I mentioned, we started using with simple 2D descriptors, then more complex 2D descriptors, like we incorporated topological, electronics, spheric, hydrophobic, and now we are using more advanced 3D descriptors also. So you will see now, when I give you a demo of operating paddle descriptors, we get data of the calculation of descriptors of about 500 to 600 descriptors. Okay. So all descriptors which are related to topology, electronics, steric effects, and hydrophobic effects, we get the huge amount of data. What are the steps of QSAR? First, we start with dataset selection, where we choose a set of chemical compounds with known activity or property values. So from where you take the data, you'll get a data either from your wet lab study or data mining. So the exercise which I'm going to take today is on the basis of the study which is done in the wet lab. It is on the chalcones compounds. Okay. So then after making a proper database of the chemical compounds with its activity value means what? It's IC50 value, 50% inhibition. I hope you understand this terminology. IC50 is the concentration of drug at which there is 50% inhibition. All right. Calculation and extraction of molecular descriptors based on this will be done, where you'll get its numerical values. Then you do descriptor or variable selection. For that, we will be using one AI-based tool known as Veka. Veka will help us to choose only the relevant descriptors. Okay. So as I told, you are going to get 600 to 700 descriptors. Of course, not all of them are important. We will be focusing on only those descriptors which are having strong correlation with the IC50 value, which is our target, right? So for that, we will use Veka. And then finally, when you get the selected descriptors, you build the model by using statistical or machine learning techniques such as regression, classification, PLS, partial least square, support vector machine, decision trees, random forest, neural networks. So I'm going to take two models for your understanding. We will do one by the conventional linear regression. We will do one by random forest. Last step is validation and evaluation for the assessment of quality of the model. And then very important and interesting application is how do you apply the model to to new or untested compounds. We call them in our language as new analogs, to predict the activity or property or to guide lead optimization. Okay, so I'm going to talk one interesting example with you so that you understand what are we actually doing, right? So suppose I want to tell you or make you understand how this model building is going to help as a big contribution to drug designing. So suppose you are working on EGFR inhibitor, right? And EGFR inhibitors, you have one very, the FDA approved and very well proven drug, which is gifatinib. Okay. So yeah, so in that, researchers actually, when take an example of EGFR inhibitor, researchers do construct a new analog, means what they do actually at the chemical level. Researcher will keep the core pharmacophore responsible for EGFR binding and making, and they make, means we are going to keep the core constant, or we will not disturb the main pharmacophore, which is responsible for EGFR binding, but we are going to make rather small rational modifications in order to improve potency, selectivity, or admit properties, which are very important for a drug. So how you can go ahead about it? So suppose we know, as I told about gefatinib, it is a very popular drug. Gefatinib has key pharmacophore as quinazoline ring, which binds to EGFR ATP site. It has N1, N3 nitrogens, which binds to hinge region by hydrogen bonding, and anilide side chain, which is involved in hydrophobic interactions. We know this. What you do when you are creating an analog, we will keep the core constant, but we will change alkyl or halogen group. You can add electron donating or withdrawing group, or you can extend or shorten the side chain. So when I'm talking about EGFR, what researchers performed or what they did was, it when gefatinib has 3-chloro, 4-fluorophenyl amino, 7-methoxy, 6-propaxy-quinazoline ring. This is the core structure. So instead of 7-methoxy, I substitute it with 7-ethoxy group. Then this is called as new analog. Why this is valid analog, I can say, because I have kept the scaffold preserved, but I have made a small modification which will improvise on lipophilicity, binding affinity, metabolic stability. So it is ideal according to me for doing its PIC50 prediction. So what I'll do, instead of making its, instead of after doing analog, after creating analog, instead of studying in the wet lab, which might or might not get successful, I will put it into the already prepared trained model for the family of gefatinib. And I'll test whether it will give me PIC50 value in the applicability domain or not. So there are some criteria which will decide whether it can be qualified as a substitute for gefatinib or not. Okay, this is the core of LBDT. So I'll give you one more example of this. When we are performing hands-on, then only I'll tell you importance of each step rather than just explaining it in terms of theory. I will give you one more interesting example of what you do. Suppose you have a research division. You are leading a research division of a pharmaceutical company focused on developing new novel therapeutic drug for migraine. Okay, migraine relief. Now, you already possess curated data set of 1,000 compounds which have very well established biological activity profiles, all of which are approved and commercially available. Right? Do you understand this situation? Now your R&D team, they come up with new library of 100 structurally diverse candidate molecules, like analogs. However, their biological activity is completely unknown. Conducting in vitro assays for all these 100 compounds, tedious, it will require substantial time and manpower, financial resources. Instead, you decide to leverage computational modeling to streamline this drug discovery process. So what will you do? You will develop QSAR model on the basis of 1,000 molecules of which you know the proven activity. You will provide a data for the software, how we are going to do today with 1,000 molecules. So that will be your training model. You use this trained model with the testing molecules also. Once you validate them with the test molecules, then you apply this already validated model with respect to the correlation coefficient, you apply this model on new analogs that your R&D team has generated. You're getting me? So those 100 analogs, you perform its prediction with the help of already proven model. So it can be linear regression, it can be RF, and then you predict the IC50 values. So if your predicted IC50 values are in the applicability domain or even better than your training set or the test set, you qualify that. So out of 100, at least 4 to 5, they will be falling under your applicability domain. You can only do synthesis of those. So then you can take it forward for docking, you can take it forward for MD simulation, you can take it forward for Swiss ADME. You're getting my point. So then we talk like clubbing or integration of LBDD and SPDD approach. So it's how much you want to explore. It's up to you, right? So this is the efficiency of mathematical modeling. You don't consume your time in R&D of new analogs by actually synthesizing it. By testing it on the model, which is AI based, you can very well decide about its prediction as a inhibitor. Okay. Descriptors. So before actually starting the exercise, I would like you to get familiar with this terminologies, what we will be often using, descriptors. Descriptors, they are basically capturing the features of molecules in terms of lipophilicity, electronic properties, hydrophobicity, molecular weight, topological features. These descriptors turns complex chemical structures into numbers, enabling statistical treatments. So when I'm saying modeling, you are building a model, of course. How can you give it a statistical treatment unless you convert it into numbers? So that is the way you have to convert it into descriptors. Now, when you are doing modeling, choice of descriptor is essential. It is very critical. Good descriptor with good data will always lead to good QSAR. Otherwise, if you have poor descriptor selection, it will cause noisy data, which can, of course, derail the mode. Algorithm of building QSAR will be focused on traditional statistical methods also, and we have machine learning methods also, like multiple linear regression, PLS, and we have SVM, KNN, neural network, RF, like this. Modern trend, which is also getting followed nowadays, is combining QACR with advanced computational techniques and large data sets to improvise on predictive power and to handle its complex in high dimensional chemical space. Now we are going to see this common algorithms in Veka, but as I told in the scheduled time, I would like to show you two models, LR and RF. So what is going to be our actual workflow? First, we will make one Excel sheet where we have a data set of the SMILE structure. I hope you understand what is SMILE. SMILE is 2D description of the molecule, which is the most validated or authentic one, accurate one. So it is simple molecular input entry line system. Always when you're doing bioinformatics, you will be asked to give 2D SMILEs. We are going to convert these two. First data set that we'll be having is your 2D SMILEs with their IC50. I'll also tell you how to convert this IC50 to PIC50. Next is structure drawing with KMSketch. So we are going to convert these SMILEs to SDF files because SDF files will be the only readable format for pattern for the calculation of descriptors. Then we will go for data pre-processing in Veka, standardization of the data, then selection of the attributes, then model building, model validation, and prediction of new analogs. Now let us start with the demo. So for the For this task, you will be needing 4 important tools. One will be ChemSketch, second will be your paddle, third will be your Veka. And yeah, these 3 are sufficient. If you want to further extend, you will also see it in Excel visualization, the result in terms of XY scatter plot. That also will show you as one example. So let us say I'm starting with this data set. So for ease of understanding, I have made separate folders. So one is our training set, one is our test set, one is our new analogs on which we have to study the prediction where we do not know the IC50 values. Test set will be different from the training set. Generally, we follow the blanket rule of 70:30 ratio. So let us say I have this training set. This is my original file, training set corrected. As I told, this comes from one wet lab study. It is about 50 molecules. It smiles IC50 and PIC50 values in micromoles. So for ease of operation and smooth understanding today, I'm going to only take 8 molecules. So I have made a separate file of it as training set corrected one. So if you see, so that it is easy for you to grasp this new concept. So always start with less data to handle so that it is better for your analysis. Then you can explore it for as much data as you want once you become expert in it. So these are 8 molecules that I have taken. This is the IC50 value. This is their PIC50 value. So you get only IC50. I have converted it to PIC50. There is one simple formula for PIC50. So I'll just show you, but I've already set it because we don't have time to calculate, right? When I take these workshops, I make people do it hands-on, everything on their own. But right now, for demo purpose, I show you how to do it, but already prepared pre-generated results or the files I'll show you. So for this PIC50, I think we all are from, all of you are from engineering background. So it will be simple for you to understand. We will put this formula 6 minus log B2 and enter. So this will be your PIC50 value. Of course, if you want to apply it to all the cells, you need not calculate for all the cells. You just put enter and it will be calculated for all. So here is how I get my PIC50 data. Okay. So always it is advisable to maintain the PIC50 data so that it is easier and it is accurate to build the model. Right? So now I have this one as my starting file or original file. Training set corrected one I'm going to use. Now we have to take this in the KMSketch, which is a very popular molecular drawing program. It is freeware. You just have to go to KMSketch and you can give your institute ID. They will give you one mail and that is how you register into this KMSketch. You can install it on your local system and you can use it. This is extremely useful in CADD, not only in LBDD. We often draw our drawings of the structures over here. Now we go to tools, generate structure from smiles, and here we are going to put one by one the smiles of this training set data. So I copy it, go to the KMSketch, paste it, and then I click on OK button. So you get this, right? Then it is always mandatory to clean the structure. So I'll clean the structure. Then I go to File and Save As, and I'll save this as molecular number 1. I'll go to the relevant folder. So I go to 2D KVCR and my folder was training set. So you have to essentially change its format to SDF and then save it. Okay. So this I showed you for your understanding. Likewise, I will convert all of them to SDF. So I have already prepared 8 SDF molecules. I'll prepare all of them in the same way, right? So once you have your SDF converted as your data set, we have to now calculate the descriptors for all these 8 molecules, which are our training molecules. So what I do is I'll go to Paddle. Paddle is also very easy to use, and it is also free software. You just have to go to Paddle descriptor. There is no installation. You just have to extract. If you have Java on your system, it will be easily operable. I open Paddle descriptor. It's a bit slow. Be a little patient with this software. Yeah. So when I open this Paddle descriptor, you can see this line, molecules directory file. You have to go here and you have to browse here your data directory where I have saved it in the 2D QSR training set. So you have to give this path. Then I go to descriptor output file and you have to give your file name over here. So you can write here Paddle training. I have already made this file, so I'm going to make it as suppose Paddle training 2. I will just to show you. So Paddle training 2, I have named it as, then I will click on save, right? Then 1D, 2D we are doing here. You can do similar way 3D. We are now seeing demo of 1D, 2D. So then I click on start button, right? Now it will tell you that descriptor calculation is completed with this average speed. You say OK, and then you go to the training set and you'll be able to see your CSV file, which is generated with the calculated descriptors. Let me show you the file. So let us open this Excel sheet. Right? So this is how you see your descriptors. Can you see there are so many descriptors over here for the 8 molecules. Now I'm going to name them because they come as autogen. You have to replace them with your numbers quickly and then you will save this file. Okay. Now these are different descriptors. All of them are for different properties. So when you say A log P, it is partition coefficient, like, right? Octanol water partition coefficient. Then you have NB, NC, number of carbon atoms, number of boron atoms, NCl. We have many, many descriptors. We are going to see only the ones which are important that we have to do through VECA. We will not understand which are important from the model building point of view. That is called as selection of relevant descriptors. And for that, you have to give input file as this training, Paddle training file. But before that, you need your target variable. What is our target variable? It is PIC50, right? So I copy this column from here and then I have to paste it in the Paddle training like this. Okay. Then you have to use this file to pre-process in the VECA to select the attributes in the VECA. Now let us open our third important tool, which is AI-based LBDD tool, and it is known as VECA. VECA also is freeware. You can install it from the website and the link. Again, you have to follow some installation steps with your registration details and you will get VECA on your system like this. Go to Explorer. It will open this GUI chooser Go to open file. Right? Now you have to load here the file which we have made. What we had made? Paddle, right? Paddle descriptor file. You have to change it to CSV. Essentially, remember, these are some troubleshooting issues which we always face. If you do not save it in proper format of CSV, you will end up in trouble. So make sure always it is in proper comma delimited format. So then what I'm going to do is I'll choose this paddle training file and open it. Okay, it must have opened in some other window. Just a minute. Yeah, so it has come over here, right? You have to remove name column. So I'll click on name and remove. Okay, now we have to follow some steps. So we go to choose, filters, unsupervised, attribute, and you standardize this data. You have to do this standardization to fit it into minimum and maximum with standard deviation. Then you remove useless. The descriptors which are not important to us will be removed. For that, I click on remove useless and apply. So we are left with only useful descriptors. Of that also, we have to select proper attributes. Once this is done, I'm going to go to this select attributes. I choose CFS subset eval. Why do we select this? There is for everything, there is a precise reason, logical reason. CFS subset eval is correlation-based feature selection. It is used in VECA to select the subset of descriptors or features for building the product predictive model. So it select only those descriptors which are highly correlated with target variable. Right? Huh? Okay. So then we choose best first. We use full training test, which we had given. You have to select PIC 50. Then you click on start button. Fine. So you can see over here, these are the selected attributes or the 2D descriptors which are important and correlated with your target variable PIC 50. We had got around 400. Out of that, we selected only this. Means tool has selected only these because they have strong correlation with your target variable. So what you have to do here is manually, you are going to note them down and you are going to make new Excel sheet and name it as SD training. So this SD training will be only of your selected descriptors. So I'll show you, I have already prepared it, but how to prepare that I will tell you. So this is my SD training file. If you open, you can observe this is the analog. These are the these are the molecules of training set. This is your PIC 50, and this you can see these are the descriptors which I've got in this VECA window, right? So I have made their Excel sheet. From where do we extract this? That I'll tell you. So we have this paddle training, right, guys? So we will go there and we will click on Control F and you have to see which parameter or which descriptor it has given. So if it is ATSC3M, I'll go to this paddle training file, type here ATSC3M, find next, right? So you will copy it, you'll make a fresh workbook of Excel and paste it in the column next to PIC 50. How I have made. Okay, we don't have time to do it one by one. So I hope I'm clear with what I'm saying and you are getting how to do it. So this is the name column where we have put ID, then comes your PIC50. So I have extracted from this paddle training only the descriptors which I have understood are relevant, right? So we have made this ST training, which is the most crucial step of VECA. Now I'll go to this VECA Explorer again. Now we have to build the model based on this. So what we do here now, go to pre-process, open file, go to ST training and open it. Right now you can see how much limited descriptors you can see in the window, only those which are relevant. Then I remove name column. You keep the descriptors. Then again you go to choose. Standardize, apply. Then you go to classify. Here we are going to choose multiple linear regression first. Use training set. More options, you have to always choose CSV because we have to get it in CSV format. I'm going to change it to PIC50 because that is our target variable. I click on start button. Fantastic. So you can see this is the table of our instances, actual values, predicted values, and error values for 8 training set molecules. And this is your summary of the result. Correlation coefficient, mean absolute error, then root mean squared error, relative absolute error, root relative squared error, and total number of instances. So you can take this data and you can plot its XY scatter also. I'll show you one example of that also, and I'll tell you meaning of this. So if you see here, the correlation coefficient is very good, 0.98. So it is very good model and it has the predicted values close to actual values. So anything which is close to +1 indicates strong correlation and strong positive result. Of course, you understand this logic. Lesser the error, the accuracy of the model is more. So you can see over here, absolute error is very less, 0.11. Root mean squared error is also less. So same is the case when you convert it percentage wise. So you can very well accept this trained model. It is showing you high accuracy. Now we also take this data and put it in the form of XY scatter. I'll show you one example for ease of understanding. I have already put it in this slide. So if you plot this XY scatter of actual and predicted values, you can very well see here that the blue points are the actual values or experimental values. Orange values are your predicted values. You can see this orange and blue are very close to each other. So that means predictions are not extremely far from actual values. The most of the points are lying between -1.5 to +1.5 for the errors. So that is acceptable, right? And the points are between 6 to 7. What is this line saying? It is the trend line. Trend line is almost a flat with slight downward slope. So that means there is strong correlation and there is no much variation between the actual and predicted. So this is how you can also confirm with the plot, but the best parameters to judge the quality of the model is your correlation coefficient and your error. Lesser the error, better is the result. More the correlation towards the +1 value, better is your result. Okay. Now, what we have to do is you have to save this model. So I'll go right click and save the model. It will be saved as your LR file, LR model file. You can save it in your 2D QSAR folder. So I have already made this. Let us say now this model. LR4. I'm naming it because I already had this previously done models. So I click on save button. Then next you have to test the data on this already made model. For that, what you do is you have to perform all these same steps on the data which is other than the training data. So new 5 molecules I have taken. If you see, this is my test set original file in the same way as we had training file. So we had 5 smiles, IC50, PIC50. From that, I will go to paddle again. I hope you are understanding and I'm extremely clear with what I'm saying. So you go to paddle and then give this SDF files. So before that, what you have to do? You have to go to Chem Sketch here and you have to give these test compounds. You will get 1, 2, 3, 4, 5 SDF files for the test compounds. You give this data to paddle. So you get now paddle test.csv. So see here, this will be your paddle.csv, which will be for 5 test molecules. Same descriptors for 5 test molecules other than the training set. From this, you don't have to go all over again to Vecca to select the attributes because we have already selected our attributes. So you just have to take this as the original file and extract the same descriptors which we had done over here, which we had done for the SD training. Remember this. SD training, we had this ATSC3M. ATSC3M is basically for the atomic mass 2D autocorrelation descriptor. MATSC is electrotopological state descriptor. Like that, everything. A log P is octanol water coefficient. So like that, every descriptor has its own significance and contribution. So are you getting my point? From the test paddle, you have to extract in the same way as I mentioned. You'll press here Control F and find the descriptor. You'll copy it and make SD test file. So let us see. This is our SD test file. Now we have to go to Vecca and now we have to load here this file. Before that, for you to understand what I'll do, it's okay even if you continue in the same window, but I'm going to delete here and freshly load the model again. So what I do over here is I go to classify, preprocess, open file, and then you are going to take here SD test file. So I go to test set, take SD test and load it. Remove name column, again standardize. Everything same on the test set as we did it for training set. Okay, then you have to, after doing this, what you'll do is, as I mentioned, there is no need to select attributes again. We are going to stick to always those attributes which we have selected in the training. So then I go to supply test set, and I open the file of SD test that we have selected. Then I go to PIC50 and more options, make sure that it is CSV once again. Then I load the model which I had saved just now. Which one we had made? LR4, right? Then I right click and say reevaluate model on the current test set. Perfect. So you get this model. Again, you get for 5 test molecules, your actual values, predicted values, error values, correlation coefficient is okay. Means for the test molecules, it is around 0.5, which is not very good, less than 0.9, but it's fine because the test set molecules are new, right? So after that, you do error also. Error error is also more. So I won't say that this model is going well with the test data, but you have tested it on the test molecules and it is moderate, I would say, not a very great model. Now we will use this model on the new analogs, the last and important step. So what we do is now, same way, we have to go to new analogs and see, this is my predictions file. Now understand, very important here to understand this step. Now, since this is new analogs, it is not having your IC50, right? It is only having your smiles, no IC50, because we are going to predict it. So you will be using this smiles. Again, the same thing. You will go to paddle. You will give SDF files. So here I have made new analog folder in which I have made 3 SDF files again. I will make paddle for this, and from this paddle CSV, which I have made as new CSV, again I will extract the same descriptors. So see here, this is my new CSV, but remember you have to make PIC50 column, but you will keep it blank. Don't forget to make it, because otherwise Veka will not understand that it has to calculate or predict the values. You have to make this blank column. So we make new.csv with PIC50 blank and calculated descriptors which are chosen again, which are same to training, which are same to test. Okay, so this is my SD new CSV with the same descriptors, if you can observe. Okay, now we go to Veka, go to pre-process, open file, go to new analogs, sdnew.csv, I have named it as, open it, remove new column, choose, standardize, apply, then you go to classify. In the supply test set, now you are going to select this new analog file. So I'll select this sdnew, close it, go to more options, make sure that it is CSV, go to PIC50, and you can keep this model. No, we have to delete this model because it is test set model. I will load training set model over here by going to training set. Yeah, we will take this LR4 model, load it, and then I'll click on reevaluate on the current test set. Perfect. Can you see? This question mark is because we do not have actual value. This is our predicted PIC50 value. So you get it's quite in a good range, -3, -3, -3. So these are the predicted PIC50 values for my new analogs. Okay. And since we do not have actual values, error is also question mark, right? So this is the most important application. Now, how do you interpret? You compare it with the training set PIC50 values, whether it is falling under the applicability domain or not, right? So I'll tell you the judgment how you actually go for the synthesis. So see, if my values of training set PIC50 are like -5, -6, and I get the new analogs with -3 range, I will select it. More negative PIC50 is cross. More positive PIC50 value is accepted, right? So If you have training set values in the range of suppose -10, -11, and if you get your predictor analog PIC50 as -12, -13, there is no sense. If you're not getting analog, which is better than before, what is the sense? But if you are getting any one analog from your new synthesized analogs, which is below -10, like -6, -7, -8, you can design them. Understood? This is how your modeling works. So now I think I showed you the multiple linear regression. Similar way, you have to just choose here from this trees random forest. So, but I think we are running lot ahead, or I means we are running late of time. I will not repeat the same thing for random forest. I think it should be clear how I perform for linear regression. Just you have to select your RF, and that will build like this. You will save in your training set your RF model, and then you execute same things on the RF model, same test set, same new analog, and see which one is giving you better predictions. All right. So this was about how to implement 2D QSAR. When you want to perform 3D also, nowadays we perform, you just have to go to paddle for a selection of 3D descriptors and use that CSV file for the model building. That becomes your 3D QSAR. All right. So this was about the application of LBDD with the demo. Now I'll go ahead with some general application points. So I've already covered this. It is helpful for virtual screening. It is used for lead optimization, for predicting activity. It is very important for toxicity and safety prediction. We use this also nowadays in combination with one more tool known as Swiss ADME, where you predict about their admit properties. Then there is one more tool, TOX2, for prediction of the toxicity. When you combine all these reports, then only you can go for further advancement into your research. Now, when I was saying you can explore and go ahead in your protocol, so if you get promising analogs, suppose all three analogs, which I have come up with the results just now with the predicted PIC50 good values, if I select all these three analogs, I will take them forward for docking. I will take it forward for MD. It's like that. That is how you standardize your drug designing pipeline, right? Now, what is the future scope for that? Now, people are also working on QSPR, which is not just limited to biological activity, but for the properties also. And so by knowing this in advance, you are getting knowledge about its regulatory use also, whether to test it for, because you know the inhibitory contribution right away, you can decide whether to go for the clinical trials or not. So the advantages are faster, cheaper, can handle extremely large libraries of compounds. So it helps in rational drug designing and useful for in silico predictions, which are not necessarily to be synthesized. Limitations, of course, is there, which are like if your model quality is not good. So like Kanika had also said, garbage and garbage relation. So if your data is not clean, it is not standardized, your choice of descriptor is not done properly, then it can go wrong. So it is always a prediction, guys. Risk of overfitting is also there, like it is good in training data but fails on new compounds. So you have to always validate by wet lab studies. Okay. Then recent trends and advances in this is you are connecting this QSAR to speed up the early stage discovery, and you're clubbing it with MN algorithms and with the automated docking tools, or automated AI driven docking and MD tools also. Conclusion, I will say it is a powerful technique, but you can't replace it. You can get a guideline. So this was all about AI and ML in LBDT. I'm just going to conclude it with the SBDD also, so that you are having a knowledge about the tools which can be used, how AI can be implemented in docking and MD also. So there is one very good tool now which has come, known as, so first if we are saying structure prediction, so as I mentioned repeatedly, you are now uh, I mean, you know this, you're familiar with this fact that you need structure when you say SBDD, structure of the receptor. So when you do not know the structure in the experiment, you use one AI driven tool known as AlphaFold, which is able to predict the structure of the receptor. So this is AI based AlphaFold. You just give your sequence, it will give you the model, it will give you the model quality depending upon the PLDDT score, and the mean error, okay, RMSE score also it will give you model to be downloaded. You use this model in the tools to get more insight about the quality of model in ProCheck. I hope you all are knowing what is Ramachandran plot. So in the ProCheck you get the plot statistics according to phi and psi angles allowable value. That decides your quality of model. So now if you are following AI driven protocol for docking, you can, and you do not have PDB file, you can build the model and that you can take forward for docking. Now when I go for, when I use AI for docking, it is one very good software known as DeepDock. DeepDock is uh, actually replacing the conventional principle of docking, which is based in traditional docking, which is physics based, which is interaction based. If you remember first a second slide which I had explained about the docking, it is completely based on protein ligand interactions. So but then this AI tool is completely based on the probability distance distribution. So in this DeepDock, it takes or learns the knowledge of protein ligand complexes to predict binding affinity more accurately, where we are making AI to operate in docking just like the Google Map. Google Map finds the best route. Here, AI in docking finds the best way how drug can bind to the protein. So I'll give a very simple example. Autodog Veena, when I say in docking, is like a student solving all possibilities and then choosing the best answer. DeepDock works like a student who has seen many solved papers and then predicts the answer directly using the pattern. Are you getting? So DeepDock is exactly one step ahead of Autodoc, where it is based on the judgment given by studying the patterns and not the actual logic of the protein and ligand interaction. So as I talked, the DeepDock is like using the AI guide how to find the best ligand poses and best way of ligand to bind to the protein. So it is not like a classical docking, which is based on empirical scoring functions. Core idea behind the DeepDock is it learns how the ligands bind the proteins by training on thousands of known protein ligand complexes. So what, when I'm saying pattern is studied, what is the pattern that is given to the model? It is the distance and orientation constraints between ligand atoms and protein residues. DeepDock will not understand the PDB or STF. Instead, it will take the data and give the pattern what it understands. It will convert the protein into the grid and ligand into the mathematical graph. Okay, so this graph will be in the display or in the presentation way, atoms as nodes and bonds as edges. So that is how it will make the model learn structural and chemical features. By using this, DeepDock builds a graph. On the basis of patterns which are given, it will give the binding site. Okay, so that is how, on the basis of the data given for previously loaded information of protein ligand, their distances. So for suppose there is one first amino acid atom carbon and its distance with the ligand's first carbon, their distance, their pattern, like this n number of atoms, this pattern is given, and that pattern is used to capture hydrogen bonding, hydrophobic contacts, and electrostatic interactions on the previously given information. And then there is distance prediction. So on the basis of this probability distribution, it will dock the ligand, it will place the ligand in the binding pocket. Are you getting? So in conventional and in computer aided, you are placing the ligand into the protein on the basis of the grid box. Here you are doing this prediction and placing the ligand on the basis of the information of their distances and pattern of interaction, right? So this step definitely ensures physically realistic positions, okay? And then the poses are ranked on the basis of predicted distance distributions, okay? And then if you want to get surety, you can load it into the PyMol and you can see the final dot complex. That is how you are replacing this AI in replacing the traditional logic of lock and key and the protein ligand interaction with the distance distribution. Okay. Next is AI and its application in simulations. So we do not have replacement of AI in simulations because we are dealing with huge amount of data. Like in if you know you have operated GROMACS, its data is trajectory data is huge, 100 nanosecond, 200 in GBs, right? You cannot give that pattern, but you can work on some parts to improvise or accelerate or guide the MD workflow. You cannot apply it for entire thing. You cannot replace because GROMACS is not a tool, it is engine. So you can just improvise on some parts according to your choice. How it can be done? You can club deep MD with the replacement of classical force fields like amber, charm. Then you can club it for the trajectory analysis also. But as I told, only some parts can be improvised, which we don't do actually, which we still trust on GROMACS, and we can just speed up the process by AI for the analysis purpose. Okay, it cannot replace the accuracy of GROMACS engine. Now, last part I would like to talk about is the case study and integration of LBDD and SPDD. How can you integrate? As we started, you do LBDD, then you can do docking of top hits that you get as best analogs, then you can do MD, then you can do admit, and then you are all sorted to go for final synthesis of the molecule. So this is the summary of tools which we discussed in this 1.5 hour. ChemSketch we studied, Paddle Descriptor, Vecca, Autodoc, GROMACS, AlphaFold, all are freewares. Okay, then as I told, how can it be AI assisted workflow? You can take AI help for deep docking. You can take AI help for trajectory analysis. And you can club it with classical docking and classical MD. Okay, this completes your drug designing pipeline with AI and ML. Thank you so much for being patient, though we ran out of time. I hope it was productive and valuable for you to give insight of how we operate in drug designing. Thank you so much. And I will now hand over to organizers for the questions and rest discussion.
Yeah, thank you, ma'am. So the session is now open to the participants. If you have any doubts, any questions, queries related to the topic, you may ask your question or you may post your question in the chat box also. There is no any question from the participants. Ma'am, let me ask you one quick question that
Yes.
basically you explained this two types of drug design, that is LBDD and SBDD. So exactly how do researchers select that which approach to be selected for while discovering the drug for a particular disease?
Yeah, that is a good question. Like I said, it depends upon the scope and objective of your work, the data set you are handling, and very important, whether you have a strong knowledge and validated target with you. If you have a validated target with you, then you can go ahead with docking MD, which is your SBDD. But you just know hypothesis that, so as I told, you have known fact that chalcones act good as cancer inhibitors. You have chalcones data, and then you come up with new analogs where you don't have the receptor, and but the model is ready, then you can go for LBDD approach. Any new analogs that you design and you do not know its biological activity, or you do not know its receptor on which it is going to act, but you have a doubt, or you have uh, you can say you have a judgment that it might act against cancer, then you go for LBDD. So basically, it is dependent on the data which you have with the confidence. Either you have your molecule structures, their analogs which you have designed, or you have validated target with you. It depends on that.
Okay, thank you, ma'am.
My pleasure.
Participants, any other questions from your side? Okay, ma'am, I think your session was too much like very detailed explanation you have given, so I don't think any more queries are there. So
Thank
let me take this opportunity to deliver the vote of thanks. So ma'am, it was a very pleasant experience to understand from your presentation that how this computer-aided drug design has become a game changer in this pharmaceutical research, which generally helps scientists and researchers to design and optimize the new drugs very faster and more efficiently. We know that generally these biotechnologists often lead the charge in discovering new biological targets and developing new biomolecules, while pharma technologists, they focus on formulation and drug delivery system and dosage. Though it is so, ma'am, you have nicely explained that how chemical engineers play a key role in further process development, then scaling up, and then manufacturing process. So it's a kind of teamwork where biotechnologists, pharma technologists, and chemical engineers all play their part of role. And Sharvari ma'am has already explained that how AI can be a new member of this team who will accelerate and strengthen the role of other three members. After listening to ma'am, I'm sure that everyone here will agree with me that why I said at the starting that she is one of the favorite trainers of participants because of her patience and teaching method. Ma'am, your presentation has certainly gave an exposure to this new era of AI-based drug development and delivery system. I'm sure that your presentation must have inspired, especially our budding engineers, to take up the career in this new field. So thank you, ma'am, for this insightful and nicely organized session. And we appreciate your for sharing your expertise with us. Thank you, ma'am.
Thank you so much, sir. It was indeed my pleasure to become part of this FDP, and thank you so much.
Yes, thank you, ma'am.
Wonderful
Good
session,
night.
Professor Sharvari.
Thank
Finally,
you.
today I could listen to your session. Last time in the workshop I missed it, but today I could.
Yeah,
It was a
I'm
wonderful
very
session.
glad that you attended it.
Yeah, so I think, ma'am, shall we end the session?
Yes, please.
Okay, okay. Okay, thank you,
I
Sharvari
would
ma'am,
like to
once again.
specially
Yeah,
thank all the participants because I mean it is too much ahead of the time. We are running late, but still everyone was patient. So I do
yes, yes.
appreciate everyone's patience also.
Yeah, so thank you all the participants for your this overwhelming response on the first day of this FDP, and then we look forward to maintain this spirit for all upcoming sessions also. So for a time being, good night everyone, and we'll meet again tomorrow at 11:30 a.m. for the fourth session of this FDP. Thank you all
Sharvari,
participants.
please share your PPT so that we can share with the participants and for the
Yes, ma'am,
reference.
sure.
Okay?
I will share. If you want,
Yeah.
I'll share this data sets also if they want to explore further. Not a problem. I will share
Yeah.
material with you. Yes.
Okay,
Yeah,
that would be great. Thank you.
Thank you, ma'am.
yeah.
Yeah.
Thank
Bye.
you. Thank you.